Hi everyone. So please welcome our next speaker, Dr. Shifra Lansky. Shifra completed her postdoc at Weill Cornell Medicine, and she has recently opened her own lab at the Weizmann Institute of Science.
Thank you. Thank you for the introduction. So one second, I'm just sharing my screen. Um, okay, does everybody see? Okay, so I'll present today the work I did during my postdoc. And during my postdoc, I was studying an ion channel called TRPV3. So TRPV3 belongs to a large superfamily of channels responsible for sensation of temperature. So basically, upon application of heat or cold or any change of temperature, these channels open up, allow ions to flow through the channel, and that this activates an action potential inside a neuron. And there are many different types of chip channels. We have chip channels that are responsible for sensation of heat, chip channels that are responsible for sensation of cold. And these chip channels have been studied very widely. So there are currently more than 250 structures of these chip channels from 20 different types of chip channels. And you could see a summary of these chip channels here. And basically what you see is that all these chip channels are tetramers. They're made of 4 different subunits. However, there's this one phenomenon that's sort of a mystery, despite almost 20 years since its discovery, and that's the pore dilation phenomenon. So this phenomenon occurs when you either activate these channels for a very long time or apply a very high concentration of agonist. And in these extreme conditions, you can see that the channels dilate over time. So they basically allow passage of small molecules in addition to the passage of ions. And so this is a mystery because when you look at the structures that have been solved so far, and like I mentioned, there are many, many structures that have been solved so far, trip channels. The pore is not wide enough to allow passage of these small molecules. So one question is how are these trip channels able to do that? So in the lab I did my postdoc in, the researcher developed the method of high-speed atomic force microscopy or high-speed AFM, which is a new type of AFM. So for those who don't know, AFM is basically a method that allows you to scan the surface of whatever sample you're imaging. So basically, you image a topography, and then you get an image. Now, normally in AFM, you get such an image in about 10 minutes, but in high-speed AFM, you get it on a sub-second and even millisecond regime. So you could get videos of proteins. So the first breakthrough in this method was here you could see myosin walking on actin on a single molecule resolution. You could see conformational dynamics of single transporters. So this is a GLTPH transporter. You could follow assembly of proteins. So basically, I hope you could see here that with high-speed FM, you could get both structural and dynamic information on a single molecule level. So it's basically quite powerful and could give you all kinds of new insights, and specifically information regarding rare and transient states. So, like I said, we studied TRPV3. So basically, we wanted to study it using high-speed FM, and the first step is to reconstitute your protein into a membrane bilayer. So you take your purified protein and transfer it into the bilayer. And then when you put it on the AFM stage, we could get these nice images. So again, to make you understand these images, these are topography images. So it's height information, where light color means high features, dark color means low features. And basically these sort of cross-like structures, these are trip channels. And in the beginning, it looks quite as expected. You see, they're all made of 4 different subunits. What was kind of surprising for us is that we could see here there are some pentamers. So molecules made of 5 subunits. So you could see them among these many molecules. You could also see them in higher resolution. And what was really cool is that you could see that these pentamers are actually reversibly formed from the tetramers. So you could see a tetramer that's quite stable for many minutes, and eventually it turns into a pentamer. You could see another example here. So a tetramer that turns into a pentamer. You could also see the opposite reaction. So you can see a pentamer that turns into a tetramer and another pentamer that turns into a tetramer. And in some cases, we could see the full transition. So we could see a tetramer that's stable for many minutes, turns into a pentamer, and then after another quite a few minutes, it turns back to tetramer. Now, finding this full reaction was a bit more difficult to find, to like detect, because you had to follow the molecule for a long time. But still, we could see at least a handful of these. So from all these events, we could get a typical lifetime for the pentamer, which was about 3 minutes. And we could not only see that these pentamers are formed, but we could also get information from the AFM how this happens. So you could actually see the monomers coming in and attacking the tetramer to form the pentamer. And here you could see also monomers dissociating from the pentamer to form the tetramer. So these are marked by these white arrowheads. You can see the monomers leaving. And if there are monomers around, then maybe there are also trimers and dimers. And indeed, in certain snapshots, we could see them. So we could see these some fragments. Now you say fragments, that's maybe doesn't sound so good, but actually we found that also these are reversible. So you could see the tetramers that seem stable, they actually momentarily dissociate and then reform, or just fragments that just diffuse around. They reform to form a stable tetramer. So based on all these data, we came up with a mechanism for how you form pentamers. So basically, you have these tetramer TRPV3 channels in your membrane. And then for some reason, which I'll talk about in a minute, they dissociate, and then monomers come, attack the tetramers to form pentamers, and then monomers dissociate from the pentamers to form again tetramers. And our next sort of thought was, well, we see these pentamers, what is its functional role? Could it be related to the pore dilated state? And the reason we thought that was, one is that a pentamer has a much larger pore than a tetramer. So you could see this pore here. And two is that pore dilation is a process that occurs on relatively long timescales, so seconds to minutes, which is also the time scale that we found for the formation of the pentamer. So what we did is we added a pore dilating agent, so this chemical that has been shown to induce formation of the pore dilated state. And we could see that when we add this pore dilating agent, we get a twofold increase in the number of pentamers and also a twofold increase in the number of fragments. And this actually sort of makes sense because you could see, according to our mechanism, in order to form pentamers, you have to first associate them. So we thought, could we check if this pore dilating agent destabilizes TRPV3? And indeed it does. So when we check the melting temperatures, we could see a significant decrease in the melting temperature upon application of this agonist. So now we think that if we look back at our mechanism, this dissociation process is a process that's favored by these agonists. So basically, they kind of enter into the subunits and probably destabilize them. So this still has to be proven. But before we continue with that, we wanted to say, okay, could we solve the structure of this pentamer by cryo-EM? This would also verify our findings with another method, and also we could see if the pore of the pentamer is indeed large enough to allow passage of small molecules. So we collected a nice data set of TRPV3, and so basically, when you just take this data set, it's very beautiful for those who appreciate cryo-EM, and then basically automatically we got a very nice structure for tetramer. So this is relatively good resolution. But if you look more carefully at your micrographs, we could see that here and there we could see pentamer molecules. So you could see them here in enlarged on the sides. And we actually succeeded in separating these pentamers from the tetramers. So this was quite a long process because these are represented in the end about 3% of the total particles. But we could see here top view. This is the first thing I got from these pentamers, but then through a long process, I was able to get more side views. and a rather low resolution map of the pentamer. But still, I was able to trace the backbone. I could see that it forms the same domains like the tetramer. So basically, it's a different oligomeric state of TRPV3. And recently, I was able to improve the resolution by finding additional side views. And now my resolution is much better. I could also trace in side chains. And we now could compare the profile of the pentamer with the profiles of the tetramer. And we could see that the profile of the pentamer, so this is the pore of the channels, and you can see the pore of the pentamer is much larger than the pore of the tetramers. And using molecular dynamic simulation, we were able to demonstrate how the pentamer is able to allow passage of small molecules. So basically, to summarize and sort of take home messages is that here I sort of showed you the first report of a pentameric trip channel over a background of more than 250 tetrameric trip channels. And that this finding was conducted through the use of high-speed FM, which I hope I convinced you is a strong complementary tool in structural biology, especially if you want to detect rare and transient states and understand their dynamics. And three is that this basically introduces a new concept, perhaps for conformational changes. So usually if you think of ion channel conformational change, you think of an opening or closing of a channel, but you don't think of a change in quaternary structure. And basically my question, one of the questions I'm kind of interested now in pursuing is to see if this could be more widespread and if basically it could be another gating mechanism for ion channels. So with that, I'd like to thank my postdoc lab, my supervisor for my postdoc, Professor Simon Schuring, and the people who helped me, and of course the funding. I'd be happy to take questions.
Thank you. Thank you, Shifra. So questions from the audience? If there are no questions, I have a question for you. Please feel free to put other questions in the chat. A very interesting talk. You work on the ion channels that respond to heat, right? So just personal interest, when you did all those amazing AFM experiments, did you expose those receptors to heat or more like agonists?
Yes, actually, one of the other goals was to expose it to heat because in AFM is done in room temperature and you could potentially also increase the heat. So we actually developed in the lab a system to increase and monitor the heat. And we did actually get some images of chip channel with the heat and we could also see some pentamers, but it was very difficult to get a lot of data because when you increase the heat, so basically you also decrease all the diffusion and all the vibrations, and it's very difficult to get high resolution images. So we were a bit struggling with that. So sort of still has to be explored. So I didn't finish that work. But yes.
If there is no more question, I can ask a follow-up question. So you mentioned the vibrations that you generate, the vibration are needed for the AFM, or the vibrations are also affecting the ion channels opening?
So when you image by AFM, you basically oscillate your tip so at the resonance frequency, and you measure basically the damping of how much the amplitude of the oscillation gets damped when you approach a surface, and you try to do this at a minimum, as minimally as possible. So you can measure the force that you apply on the surface, and you always make sure to make it as low as possible. And of course, you have controls to make sure that you're not ruining your sample. And we know what is the force that is sort of good for the protein.
Yeah. Thank you. I see here a question by Abigail.
Hi, thank you. Really cool technique. I'm really not an expert in these techniques, so I was wondering what are the major limitations of it? Like, could you only image things over a certain size, under a certain size, or are there
Yeah.
other things to consider?
Yes. So I think one of the main well, there are two limitations to the technique. One is the resolution. So, I mean, with this protein, we were able to see very nice stuff, but it's not atomic resolution. So it's not cryo-EM resolution, it's not crystallography resolution. So basically, the resolution is more nanometric, XY, though on the Z scales actually could get to angstrom resolution. But that's one major hurdle that we have to overcome. So basically, if you want to get atomic resolution information, I would say you couple it together with another atomic resolution technique. And another limitation is that this is a surface method, so it has to be on the surface. So it doesn't monitor what happens in the bulk. In the case of membrane proteins, therefore, I think this is a super good method because membrane proteins are anyway sort of on a bilayer on the 2D, so there's not really that issue, and you could just put these vesicles with the membranes on the surface without any tethering. You don't need to tag it, and you could see that these molecules diffuse naturally. So it kind of is a nice setup. But for soluble proteins, you could also study, but you need to think how to do it in such a way that you don't disrupt your biological question.
All right. Thank you so much, Shifra, for your fascinating talk and work. And thank you for the question